Hi, and welcome to this series exploring landmark experiments in protein science based on my book of the same name, which can be purchased on Amazon, and that link is in the description. In the last video, I talked about glycolysis and the Krebs cycle, which are pathways where precursors required to make energy are generated. And while glycolysis makes a little bit of energy in the form of adenosine triphosphate, or ATP, by substrate-level phosphorylation, which is when phosphate is added to ADP as a result of a purely enzymatic reaction, most of the energy used by cells is generated by oxidative phosphorylation, in which molecules generated in redox reactions of the Krebs cycle, like NADH and FADH2, provide electrons and protons that drive the formation of ATP. And so in this video, I'll talk about the mechanism by which these molecules do this. In the early 1930s, Otto Warburg identified a new class of molecules involved in the transfer of hydrogen ions, which he named flavoproteins, the first of which was the so-called yellow enzyme, since it changed from a colorless molecule to a yellow color when oxidized. Yellow enzyme was later renamed lumeflavin. Around the same time, he also isolated another flavoprotein which came in two forms: nicotinamide adenine dinucleotide and nicotinamide adenine dinucleotide phosphate, NAD and NADP, the former of which is reduced to NADH in the Krebs cycle and is used to generate ATP. And following claims that more phosphate was esterified, which means that the phosphate is attached to another molecule, during the Krebs cycle than could be accounted for by substrate-level phosphorylation, it was suggested that phosphorylation was somehow coupled to electron transfer. However, attempts at showing that this was the case had failed. So in the late 1940s, Friedkin and Lehninger investigated the possibility that phosphorylation was directly linked to redox reactions, reasoning that the demonstration of coupled phosphorylation in these preparations by measuring net uptake of inorganic phosphate has not been possible, since the necessary transphosphorylase is required to cause phosphorylation of acceptors such as glucose, creatine, etc. via adenosine triphosphate, or ATP, have been removed in the washing procedure. However, it is readily possible to demonstrate the maintenance of the esterified phosphate of ATP coupled to oxidation. In the absence of oxidizable substrate, ATP is quickly dephosphorylated by phosphatases, which are enzymes that dephosphorylate proteins. In the presence of active oxidation of added substrates of the Krebs cycle, the level of esterified phosphate may be maintained at or somewhat below the starting level. The use of inorganic phosphate labeled with P32 provides proof that such maintenance is a dynamic resultant of dephosphorylation and oxidation-coupled rephosphorylation, since after oxidation has taken place, a large part of the P32 is found in the esterified phosphate fraction. And so P32 is a radioactive phosphorus, and molecules that incorporate it can be detected and distinguished from those that are not radioactive using a Geiger counter. So Friedkin and Lehninger measured the amount of esterified phosphate in rat liver homogenates to determine if phosphorylation was coupled to electron transport between NAD or NADP and oxygen. And so these are the different components of the Krebs cycle that were added to the liver extracts, and these columns show the oxygen uptake, the inorganic or free phosphate, the total esterified phosphate, and the esterified P32 phosphate. At time 0, we can see how much of each is present, and then how much phosphate is returned to inorganic phosphate in the absence of added components due to phosphatases, as shown by a decrease in esterified phosphate and an increase in inorganic phosphate after 4 minutes incubation. However, it can be seen that the oxidation of all Krebs cycle intermediates tested caused some degree of maintenance of levels of esterified phosphate compared to the level without substrate, and we see this by the fact that all esterified phosphate remains well above 158 and close to 302. The data also show that oxidation of these substrates causes substantial incorporation of inorganic P32 into the esterified fraction, ranging from 10 to 39% of the P32 originally added as inorganic phosphate. They also found that cytochrome C, which I talked about in the last video and is a protein shown to be involved in redox reactions, was required for this process. And so here they incubated ATPase, cytochrome C, ATP, and P32 in the presence of various molecules capable of reducing cytochrome C and assessed how much oxygen uptake in esterified P32 there was in the presence of these molecules. And here malate was used as a positive control for phosphate esterification. And so they found that although there was an extensive flow of electrons over the cytochrome system as indicated by the oxygen uptake when cytochrome C was reduced by these reagents, there was no esterification of inorganic phosphate except in the case of ascorbic acid, which showed a small incorporation. This indicated to them that esterification occurred after the reduction of cytochrome C by NADH. Importantly, the authors found that ADP was the only molecule that could act as a phosphate acceptor. Around the same time, in the mid-1940s, electron microscopy, which is a microscope that uses an electron beam instead of light to look at samples, was well enough developed to enable Albert Claude to determine that succin oxidase and cytochrome oxidase were located in mitochondria, and in the 1950s, George Palade described these new structures like this. The outer membrane can be considered as the limiting membrane of the mitochondrion. The inner membrane is connected with a characteristic structural detail described under the name of cristae mitochondrialis. Two spaces or chambers are outlined by the mitochondrial membranes, an outer chamber contained between the two membranes and an inner chamber bound by the inner membrane. The inner membrane is penetrated and, in most cases, incompletely partitioned by laminated structures which are anchored with their bases in the inner membrane and terminate in a free margin after projecting more or less deeply inside the mitochondrion. In the late 1950s, a mitochondrial fraction of animal tissues was found to be required for the generation of ATP by oxidative phosphorylation, and Raker and his group were able to further separate it into two fractions. The particulate fraction catalyzed electron transport without concomitant phosphorylation. The addition of the soluble component to the particulate fraction restored oxidative phosphorylation. During early attempts to purify the soluble component, which they called coupling factor, it was observed that a magnesium-dependent dinitrophenol-stimulated ATPase was purified together with the phosphorylation activity. Characterization and further purification of the ATPase was, therefore, undertaken in order to evaluate its relation to the coupling factor. And so they incubated ATP, magnesium, and the enzyme, so ATPase, together for 10 minutes and assessed how much ATP, ADP, AMP, and inorganic phosphate were left. And before looking at the results, I'll remind you that ATP has 3 phosphates, ADP has 2 phosphates, and AMP has only 1. And so you can see that although the same amount of ATP was added in both cases, the addition of DNP decreased ATP and increased ADP and inorganic phosphate. And notice how no AMP was formed, which indicated that the enzyme only hydrolyzed 1 phosphate from ATP. And this experiment shows the dependence of ATPase on divalent cations, which are elements that are missing 2 electrons, and in particular magnesium and cobalt, as shown by the increase in phosphate in the presence of DNP. Interestingly, they also showed that ATPase could operate in the reverse direction. Here we see that increasing the amount of soluble factor to the particulate fraction resulted in the concomitant increase in phosphate uptake, which was completely inhibited by the addition of DNP, since, as we saw earlier, DNP causes the hydrolysis of ATP. They also wanted to confirm that it was ADP that was phosphorylated in this reaction, so they repeated their experiments using radioactive phosphate, so P32, and found that ATP-containing phosphate 32 increased significantly with addition of coupling factor. And so these results suggested that a single enzyme was responsible for the hydrolysis of ATP into ADP and phosphate, and for the synthesis of ATP from ADP and phosphate, and confirmed that ATPase was the coupling factor. However, since they found that the coupling factor was insensitive to oligomycin, an antibiotic known to inhibit oxidative phosphorylation and ATPase activity, they looked for another protein that might mediate this sensitivity. A few years later, in the early 1960s, they found that treating mitochondrial particles with trypsin and urea isolated what they called the TU particle for trypsin and urea, which they found conferred oligomycin sensitivity to ATPase. And here we see the results of these experiments. As expected, ATPase is not sensitive to oligomycin, and we see this since phosphate is cleaved from ATP in the presence of oligomycin. But adding TU particle to ATPase makes the ATPase sensitive to oligomycin, and we see that here by the fact that 1.54 micromolar phosphate is cleaved without oligomycin, but only 0.3 micromolar is cleaved in the presence of oligomycin. And so they called the factor responsible for this activity oligomycin sensitive factor, FO. They also found that if the precipitation of FO and ATPase was carried out in the presence of 1.65 micrograms of oligomycin, the precipitated complex had only 25% of the activity of the control complex prepared in the absence of oligomycin. This indicated a rather firm attachment of oligomycin to FO in the presence of ATPase. Around the same time, Griffith's group was able to isolate from mitochondria, prepared from beef heart, 4 primary enzymic complexes that are operative in the oxidation of succinate and reduced diphosphopyridine nucleotide, or DPNH, which we now call NADPH, in such a manner that their ability to recombine with appropriate partners in the overall sequence of electron transfer has been retained. And these are the complexes in question. In complex 1, NADPH reduces coenzyme Q, whereas succinate reduces it in complex 2. Reduced enzyme Q in turn reduces cytochrome C in complex 3, and notice the name change from ferri to ferrocytochrome C. And finally, cytochrome C is oxidized by oxygen in complex 4. However, the authors emphasize that reconstitution, as described in these studies, is not merely the result of coupling 2 or more reactions by means of a common water-soluble intermediate. Unlike systems interacting by coupling, it is necessary for the sum of the reactions to occur that the enzyme systems in question be mixed together at high concentration prior to assay. Once reconstitution is achieved in this manner, the mixture may then be diluted and assayed at an appropriate concentration. And they noted that this would be unnecessary if reconstitution of activity was a simple consequence of the fact that certain enzymes that act sequentially are present together in the assay medium. In general, simple enzymic coupling of one reaction to another is easily achieved with either soluble or particulate enzymes. And so in these experiments, they found that cytochrome C was efficiently reduced by NADPH when they mixed complexes 1 and 3 together, whereas it was reduced by succinate when complexes 2 and 3 were combined. And both systems were activated when all 3 components were incubated together, and these were both inhibited by animycin A, but only the NADPH cytochrome C reaction was inhibited by amytal. These same combinations were sufficient to reduce oxygen if complex 4 and cytochrome C were included, and the italicized conditions were those in which the enzymes were mixed at high concentrations, whereas the other conditions weren't. And here we can see that both the succinate and the NADPH activities were high when the entire system is reconstituted. And again, animycin A inhibited both reactions, whereas amytal only inhibited the NADPH activity. These experiments indicated that the 4 complexes of the ETC needed to be arranged in a specific sequence to function, and the authors provided this diagram to help visualize the process. NADPH provides electrons to complex 1, and succinate provides them to complex 2, and both transfer their electrons to complex 3, which transfers them to cytochrome C, to complex 4, and finally to oxygen. In the mid-1960s, Fernandez-Moran and his group reported that, by careful comminution of beef heart mitochondria in a sucrose medium, well-defined fractions were prepared, one of which, the ETP, contains particles with an essentially intact electron transfer chain, divested of the primary dehydrogenating enzyme complexes. The capacity for both electron transfer and oxidative phosphorylation was retained in an analogous particle, the ETPH. They confirmed that the ETPH was composed of flavins, cytochromes, iron, copper, coenzyme Q, phospholipids, as well as proteins, and obtained the first clear images of the ATPase in mitochondria, which revealed a characteristic polyhedral or round structural unit 80 to 100 angstroms in diameter as a basic component of mitochondrial membranes. The vast numbers of these particles, as well as their regularity and periodicity, are the most remarkable features. This repeating structural entity was designated the elementary particle, EP. If a fairly uniform distribution is assumed, the total number would be of the order of 10,000 to 100,000 elementary particles per mitochondrion, depending on the size and the type of mitochondrion. As best seen in paired arrays along a crista, each elementary particle consists of the following 3 recognizable components: a headpiece, a stern or stalk, and a base piece, which is the region of attachment of the stalk to the crista. The headpiece is the most conspicuous portion, being polyhedral and asymmetric by virtue of the stem region. The double membrane structure was preserved in specimens of ETPH, which was the fraction with ATPase activity, whereas only a single membrane structure was observed in the ETP. And based on these images and the research mentioned above, it was determined that Raker's coupling factor, now called the F1, was the headpiece and stalk of the ATPase, whereas the FO subunit was the membrane-bound portion. And while all this was going on, Peter Mitchell suggested the chemiosmotic hypothesis in 1961, in which molecules created during the Krebs cycle were used to create a proton gradient between the inside and the outside of mitochondria by using translocation systems to move hydroxide ions and protons to either side of the membrane, thereby creating an excess of protons on the outside, or in the cytoplasm, and an excess of hydroxide ions on the inside of mitochondria. And this gradient was proposed to supply the energy for phosphorylation of ADP to generate ATP. As Mitchell explained, according to the chemiosmotic coupling hypothesis, the differential of the electrochemical activity of the hydrogen and hydroxyl ions across the membrane, generated by electron transport, causes the specific translocation of hydroxyl and hydrogen ions from the active center of the so-called ATPase system, thus effectively dehydrating ADP and phosphate. The underlying thesis of the hypothesis put forward here is that if the processes that we call metabolism and transport represent events in a sequence, not only can metabolism be the cause of transport, but also transport can be the cause of metabolism. Thus, we might be inclined to recognize that transport and metabolism, as usually understood by biochemists, may be conceived advantageously as different aspects of one and the same process of vectorial metabolism. In the mid-1960s, Paul Boyer started thinking about how enzymes undergo conformational changes, that is, how the three-dimensional shape of a protein can change during enzymatic reactions, and proposed the conformational hypothesis, which suggested that the energy required for phosphate transfer to ADP could be supplied by conformational changes in the ATPase protein complex. Boyer and others developed this hypothesis over 20 years and eventually merged it with Mitchell's chemiosmotic hypothesis, which accurately described the mechanism of ATP production. The rest of this video and the next will be dedicated to this topic. Evidence for Mitchell's chemiosmotic theory started to arise in the mid-1960s when Jagendorf and his group, who were studying chloroplasts, a plant organelle that converts light into sugars, which can then be used to generate ATP in mitochondria, showed the existence of a high-energy condition of isolated chloroplasts caused by illumination in the absence of phosphate or ADP. This state is inferred from the ability to form ATP in the post-illumination darkness. Accompanying the condition is an apparent uptake of hydrogen ions. A feasible interpretation stemming from the chemiosmotic hypothesis for the mechanism of phosphorylation in double membrane-containing organelles, like chloroplasts and mitochondria, is that illumination causes the uptake of hydrogen ions into the inner space of the granite disk double membranes. The resulting inequality in hydrogen ion electrochemical activity across the membrane is postulated to be, in itself, the high-energy condition able to drive the formation of ATP. If this interpretation is correct, the same high-energy condition should be formed artificially, entirely in the dark and without electron transport, by loading the inner space of the granite disk membrane with protons. The authors showed that this was indeed the case in the following experiment, but before I show it I should mention that pH is a ratio of protons and hydroxide ions. The presence of an equal number of these ions has a pH of 7, and the pH decreases as more protons are added, and so a pH of 2 is very acidic and has a lot more protons than hydroxide ions. And conversely, it increases as protons are removed, or by adding hydroxide ions, and in this case a pH of 12 is very high. And here we see the results of these experiments. In the first row, chloroplasts are bathed in an acid bath and ATP formation was assessed in a basic bath at a pH of 8. And we see that in this case, ATP was formed. However, ATP formation was lost if the acid bath was followed by a basic bath at pH 6.5. The reversibility of this system was shown in row 3 by returning the chloroplasts to an acid bath after the basic bath, where we see that ATP was once again formed, noting that different acids changed how much ATP was produced. They also showed that the efficiency of the system was dependent on the pH at each stage. Here, the x-axis is the pH of the acid stage and the y-axis is the ATP produced. And we see that the lower the acid stage, the more ATP is produced. And we also see that more ATP is produced the higher the pH is when ATP formation is assessed, although it's optimal under about 8.5. And this can be better seen in this graph where ATP formation increases with an increase in pH, again plateauing at about 8.5. Furthermore, formation of ATP seems to depend not only on the pH in each stage, but also to a large extent on the actual size of the difference in pH between the two stages. The minimal pH difference for synthesis of measurable ATP ranges from 2.2 to 2.9 units, and the optimum from 4 to 4.5 units. And this is shown in this experiment, where an acid stage at a pH of 3.8 requires an ADP stage of about 8.3 for optimal ATP synthesis, whereas the basic stage needs to be above 9 for ATP production to be optimal when the acid stage is increased to 5.2. The authors concluded that the results are, on the whole, consistent with the chemiosmotic hypothesis for the mechanism of phosphorylation, as elaborated by Peter Mitchell. In this model, account is taken of the inner and outer regions of the granite disk membranes. When placed in acid, protons would be expected to penetrate the interior space depending on the external pH. Presumably, cations would be simultaneously displaced to preserve electrical neutrality. Added organic acids could penetrate in the undissociated form without the need to expel cations and provide an internal reservoir of dissociable protons, thereby accounting for the higher yields. On raising the external pH to 8 or more, a proton gradient would be present from inside to outside, and this in itself would have thermodynamic potential. The height of the potential would depend on the ratio of pHs inside and outside, and this could account for the requirement of a distinct pH difference between the acid stage and the phosphorylation reaction. Indeed, the alkaline displacement of the phosphorylation reaction pH curve due to raising the acid stage pH was one of the predictions of this model, and the experiments were performed in order to test this. The actual yield of ATP at any pH probably reflects a balance between the pH dependence on the relevant chemical reactions and the thermodynamic effect, which is a matter of the change in pH from the previous acid stage. Given a pH differential across the granite disk membrane, its potential might be translated into ATP formation via an anisotropic, membrane-bound reversible ATPase as discussed by Mitchell. Peter Mitchell's group came to similar results using rat liver mitochondria, except that mitochondria first had to be bathed in an alkaline solution and ATP measured in an acid bath, noting that, from the point of view of the chemiosmotic hypothesis, it is especially interesting that our observations indicate that ATP synthesis in rat liver mitochondria is associated with the passage of protons inwards as predicted, whereas the synthesis of ATP in chloroplasts is associated with the passage of protons outwards. This observation is in keeping with other evidence, which implies that the polarity of the ATPase system in mitochondria is opposite to that of chloroplasts. Our findings would thus seem to help to promote the status of the chemiosmotic hypothesis towards that of a theory. And so to summarize, molecules produced during the Krebs cycle donate electrons to the electron transport chain in mitochondria, which is composed of 4 independent protein complexes and cytochrome C. NADH donates electrons to complex 1, and succinic acid and FADH2 donate electrons to complex 2, both of which transfer their electrons to coenzyme Q, then complex 3, cytochrome C, and finally to complex 4. In this process, each electron transferred from one complex to the next induces the translocation of protons from the inside of mitochondria to the inner membrane space, so the area between the two phospholipid bilayers, thus generating a proton gradient across the inner membrane. In the last complex, the electrons are transferred to oxygen along with 2 protons, resulting in the formation of water. And so this brings this video to an end. I hope you enjoyed it. And if you did enjoy it and would like to know more about this topic, please check out my book, Landmark Experiments in Protein Science, using the link in the description. And if you have any questions, please leave them in the comments and I'll do my best to answer them. And I hope you'll join me for the next video on the mechanism by which the ATPase attaches phosphate to ADP to form ATP.